Introduction. Background music fades in voiceover. Welcome to the world of molecular biology and the fascinating topic of antibiotic resistance. In this script, we will delve into the intricate mechanisms behind antibiotic resistance, exploring various molecular components and processes that contribute to this global issue. So let's get started. Background music fades out. Animation showing horizontal gene transfer between bacterial cells. Voiceover. In summary, the emergence of beta-lactam resistance is driven by the production of beta-lactamases, enzymes that modify or degrade beta-lactam antibiotics. This resistance can be further amplified through horizontal gene transfer. Efflux pumps are vital in antibiotic resistance as they expel antibiotics from bacterial cells, preventing their action. These pumps are diverse and can transport a wide array of antibiotics. They are regulated by intricate networks, making them highly effective in conferring resistance. Scene. Visualization of bacterial cell membranes. Voiceover. The permeability of bacterial cell membranes plays a crucial role in antibiotic resistance. Alterations in the membrane structure or composition can limit the entry of antibiotics into the cell, reducing their efficacy. Animation showing altered membrane permeability. Voiceover. Bacteria can modulate membrane permeability through various mechanisms, such as the modification of membrane lipids or the upregulation of efflux pumps. These adaptations contribute significantly to antibiotic resistance, making it challenging for antibiotics to penetrate the bacterial cell. Voiceover. Close-up of DNA gyrase/topoisomerase mutations. Voiceover. These mutations result in a decreased binding affinity between the quinolones and the enzymes, preventing the drugs from inhibiting DNA replication effectively. This allows bacteria to survive and multiply despite quinolone treatment. Scene. Diagram of enzymatic modification of antibiotics. Voiceover. Bacteria have developed sophisticated enzymatic mechanisms to modify existing antibiotics, rendering them ineffective. These modifications include the addition or removal of functional groups or chemical moieties from the antibiotic molecule. Animation showing enzymatic modification. Voiceover. Enzymes involved in antibiotic modification include acetyltransferases, phosphorylases, and nucleotidyltransferases, among others. These modifications can substantially alter the antibiotic structure and prevent it from binding its target, thus conferring resistance. Together, transposons and plasmids facilitate the spread of antibiotic resistance genes in bacteria. Transposons can relocate within or between genomes, carrying resistance genes. Similarly, plasmids are DNA elements that can be transferred between bacteria, enabling the horizontal transfer of resistance genes. Scene. Molecular representation of antibiotic binding to a target. Voiceover. Bacteria have developed mechanisms to modify the target sites of antibiotics, making them less susceptible to the drug's action. This can occur through mutations in the target protein or changes in its expression level. Animation showing antibiotic target modification. Voiceover. Examples of target modifications include changes in the ribosomal subunits, altering the binding affinity of antibiotics such as macrolides and aminoglycosides. These modifications prevent the antibiotics from effectively interfering with bacterial protein synthesis. Scene. Molecular representation of different antibiotics. Voiceover. Macrolides, tetracyclines, and aminoglycosides are well-known classes of antibiotics. However, bacteria have developed various strategies to resist their antimicrobial activity. Animation showing mechanisms of resistance to macrolides, tetracyclines, and aminoglycosides. Voiceover. Some of these mechanisms include efflux pump-mediated resistance, enzymatic modification, ribosomal protection proteins, and target site modifications. These adaptations enable bacteria to evade the effects of these commonly used antibiotics. Scene. Visualization of bacterial biofilms. Voiceover. Biofilms are complex microbial communities encased in a self-produced extracellular matrix. Bacteria within biofilms exhibit increased resistance to antibiotics compared to planktonic cells. Animation showing antibiotic resistance in biofilms. Voiceover. The extracellular matrix of biofilms provides physical protection, preventing the penetration of antibiotics. Additionally, the altered gene expression and metabolic activity of bacteria within biofilms contribute to antibiotic resistance, further complicating treatment strategies. Scene. Diagram of two antibiotics working together. Voiceover. Synergism and combination therapy have proven effective in combating antibiotic resistance. By combining multiple antibiotics or using drugs that enhance the activity of existing antibiotics, we can overcome resistance mechanisms and improve treatment outcomes. Animation showing antibiotic synergism and combination therapy. Voiceover. Synergistic interactions between antibiotics can enhance their individual effectiveness, inhibiting multiple resistance mechanisms simultaneously. Additionally, drugs that inhibit bacterial resistance mechanisms can improve the efficacy of existing antibiotics, providing more potent treatment options. Horizontal gene transfer is a key factor in the development of antibiotic resistance. Bacteria can transfer antibiotic resistance genes to others through processes like conjugation, transformation, and transduction. This rapid transfer contributes to the emergence and spread of antibiotic-resistant strains, emphasizing the need for careful antibiotic use and infection control. Antibiotic resistance monitoring involves data collection and analysis on bacteria's susceptibility to different antibiotics. Molecular techniques like PCR and whole genome sequencing detect resistance genes and genetic variations, providing insights into transmission and evolution of resistant strains. Monitoring helps understand resistance dynamics and develop effective preservation strategies. Understanding the complex molecular mechanisms of antibiotic resistance is essential to address this global problem. Genetic, enzymatic, and physiological factors drive resistance evolution and transmission. Let's work together to protect human health and maintain the effectiveness of antibiotics. Thank you for joining this exploration of antibiotic resistance.